Good morning, everybody. First of all, as you heard, the webinar will be recorded. So please turn off your microphone. The camera is optional. It's up to you if you want to be recorded. Remember that now we have the program with different presentation, and we will have a final discussion. So you can post your question on the chat. So to promote the discussion at the head of the different presentation. I'm Bruno Mettetti, so we have a little bit of code, so my voice is a little strange. I'm the coordinator of the BreedingValue project. The BreedingValue is a NAU project, it's a Horizon 2020 project. The BreedingValue started almost 4 years ago. And we are going to the end. We had an extension, and the end of the project will be next June 2025. The project included, it's a very, maybe most of you already know the project, but for the new people, I'll give just a few indications. It includes 20 partners, including 7 small and medium enterprise, from different 8 countries. Thanks to open calls, then we have included other 10 subcontractors that in some way, most of them are again new small and medium enterprise that have been in some way included in the project. We are the coordinator. The budget, total budget was almost of €7 million. And I say that the duration is not now 48 months, but 54 months. The project is focused on 3 major berries: strawberry, blueberry, and raspberry. The main aim of the project, and this is really very, very synthetic. So then after I will leave the floor to my colleagues that will go more in deep on the description of specific tools that have been generated at the Germinate platform. The reason of the Germinate platform was defined for the project as an important tool finalized to store, analyze, and disseminate all the data that have been generated by the project. The project has a specific aim to provide and characterize and selection of the superior genetic resources, develop new tools for genotyping and phenotyping the genetic resources with the aim to determine the resilience and the fluid quality of the cultivars and of the breeding material. So in the overall, the project has a main aim to increase the value of the AU berry genetic resources. This work has been organized in the activity of 4 major research and development work packages. The first one finalized to characterize the genetic resources, identifying different accessions from the germplasm bank that have been in some way involved in this project, and then breeding material and population provided by different breeding programs, private and public breeding programs, and the organization of specific field trials in different environmental conditions and cultivation system with the aim to characterize this genetic material and identify genotypes with specific important traits to be used in further step of the breeding program. Combined, this work was also combined to the application of different genotyping tools, mostly for the identification and validation of new markers associated to important traits, to apply almost for the first time on berries of strawberry and raspberry in particular, new tools like genomic selection, and demonstrate the powerful of this technology for improving the capacity of the breeding program in berries, and identify specific, say, markets that associated are important traits. So again, to be applied in the traditional breeding. To the genotyping technology have been combined new phenotype technology, specifically related to identification of higher fruit quality and plant resilience. Plant architecture, it's an important issue related to the fact that for the different type of plant, it's real that for the different type of berry crops, it's important to identify the right plant for and the programming of the plant in order to promote and expand the cultivation season in different climatic conditions. The phenotyping has been developed applying also different new technology based on image analysis, metabolomic studies, and more important for the consumers, the concept of panel test, sensory test, so to better characterize the sensorial characteristic of the different new genetic product. And then finally, thinking to the need to transfer, identify and transfer to the consumer the most appropriate genetic material with more identified and requested by the consumer type of fruit, were developed different tools for identify what are the consumer demands. So a specific survey and focus group were organized, defined, and the sensory profile of the different berry were identified, and several consumer tests have been organized for testing different genetic material in different conditions, in different countries, so with the participation of different type of consumer. All this activity has generated a very large quantity of data from the analysis of genetic resources, from the genotyping study, from the phenotyping study, from the consumer side study. So this is the reason why, since the beginning of the project, we have identified the need to create a new database to collect all this information, to collect all this data, and so to make it as much as possible public and accessible to all breeders, to all institutions that have an interest to know what we were able to generate with this project. So today, this webinar has the main aim to introduce you, to present you the Germinate platform that specifically has been organized by colleagues from the James Hutton Institute in Scotland, and in collaboration with Bjorn Usadel and Elisa Schenger from Germany, that they are coordinating the work package 5 related to the data management and analysis. This is one of the last event related to the project. I want to already anticipate all connected people that please mark in your calendar that there will be another important event that will be on site. It will be in Italy, in Ancona, the first week of April, including the final workshop introducing and disseminating all the information, major results that are being achieved during the period of the project. So I invite you to remain connected with us, and possible to follow also this last event expected for the next year. So I stop here my presentation, and I ask my colleague to continue now with the program. Stop sharing my presentation. Okay.
Yeah, so it'll be Elisa next.
Yes. So yes, Elisa.
Yeah, it's me. Do you see this now?
Yes. Yes.
Yeah. Okay.
No.
So this is prepared by Lisa mostly, and I'm presenting now a short introduction into why data management is important. especially for our berry data in our project. Of course, berry production and the BreedingValue project is dealing a lot with European or international germplasm. And we are looking into resilience of the cultivars as well as breeding and pre-breeding material. We want to look at the fruit quality of cultivars as well as breeding and pre-breeding material that includes metabolomic data as well as organoleptic qualities. And you already start seeing that if you look at many of these different things, we actually have to link them somehow so that we can actually have a characterization and selection of superior genetic resources. So that in the end, we have breeding tools, marker arrays, sensor shows, and selection models that will help us selecting and improving berry germplasm. Therefore, in BreedingValue, as Bruno has already pointed out, so he has given a fantastic introduction already, we actually have a lot of different things. We're working with germplasms, genomics, fingerprinting. We have a lot of trials where we look at weather data, phenotypic data, and bring this all together. But we also have metabolomics because we need to know what kind of quantitative traits do we have that we can measure and quantify objectively. At the same time, we have panel tests, which actually ask for subjective or semi-objective quality by consumers. It doesn't help us if we have a lot of metabolites in here which are supposed to taste sweet and nice, if our panel tests say, well, these strawberries don't taste nice, or these blueberries are actually horrible. So we have to bring this together. And in addition, we have surveys. As you see, bridging from surveys all the way back to germplasm, there's an absolute need to bring this data together and actually link this data. So we have also established across our partner, remember this is a pan-European project, data collection protocols, defined in each work package and helped by the work package leaders. We're working on metadata, harmonized metadata documentation. Is the data actually meaningful? Is it well annotated? Do we even have the metadata? What could that mean? Well, how was the weather like? How were the plants grown? How were the plants treated? And what kind of metadata we need? Luckily for us, of course, we are not standing there alone. But for example, there is this standard MyAP. This is a minimum information about plant phenotyping experiments in the broad sense, comprising also RNA-seq and metabolomics data, so that we actually know what kind of data to have and to collect. Nevertheless, getting all and sufficient metadata is not always easy. In different experimental stations in different countries, different metadata might be important, and it might not always be possible to get everything. So one really has to be sure to make this possible, because that increases reusability, but also helps other working groups to use your projects later on, and of course cite your projects if it's published. It increases the longevity of the data, of course. And it helps us to increase the analytic potential because we can bridge different data sets. For example, does a specific metabolite influence taste? What are the underlying QTLs? What are the underlying genomic regions causing this? For that, of course, we need to have standardized protocols and data templates, which have been established in BreedingValue by Elisa and Paul. We need to use a common language, well, for example, English, but we also need to speak the common formal language that machines can understand and that it can actually go into our databases. And the database understands that this and this is not the same. So we have to additional parameters and have to have good data structures that support this. So that means, once again, we had to develop and have developed harmonized data template files that can then be used and are being used across our consortium for uploading into the Germinate database, so that when somebody in Spain does an experiment and says we grew strawberry under these conditions, and somebody in Turkey or in Norway does the same and actually says, well, we also grew strawberry under these standard conditions. We know, for example, it's both strawberry that is important, but also the conditions. So we look at these harmonized protocols which have been established for data and sample collection, because as you can imagine, if you harvest from the vine, if you harvest in a different way, or if you just have to store the berries first somewhere, then of course they degrade. That's common knowledge. But how long can you store them? How do you harvest them? This has all been established. Also with our good friends like Sonja Osorio and Klaus Olbricht, who have very good experience in how to do that. So then we need to maintain consistency as a challenge. We use the same trait names, scales, units, and intervals. This sounds very simple, but even if you look at the scientific name, well, this could be a good one. This is the real one, of course, and this is the official abbreviation, but maybe you forget the space here. And for a machine, that can be difficult. We have to use the same names and even identifiers for our cultivars, genetic resources, and sample material. And that was a major work to actually identify what kind of genetic resources we have in Europe, what kind of cultivars, and bringing them together. And I can tell you, it's a lot. Finally, we actually have to look and use the same file formats that data is consistent. Finally, there is, of course, always a thing that certain data needs to be restricted. We might have legal aspects. There might be interest, of course, by breeding companies that the data has to be kept confidential. We have to have a publication first model. This is also implemented in the Germinate platform. And of course, we're talking about the public data today with you, which is already quite substantial. So, of course, we need to also keep this in mind. And even if data is public, of course, it might be that you're working with commercial cultivars. So it might work in different countries. Also, of course, most of the data has to be is often published first, so you can read about this, and I actually have it nicely described. But much data, as you will see, is available now. So what were the challenges which have been overcome by Elisa, Paul, and Sebastian? A clear definition of protocols, templates, of course, before everything started. It doesn't help you if you start something and then afterwards you learned you had to do it in a different way, then it's too late. You all know this if you work in the field. A clear communication, training materials, brief training materials, and training again, because of course one forgets, and if it's not the exact same procedure as working in this lab, it can change. Follow-ups and immediate feedback. Of course, the data was checked by Elisa and Paul, that the data is actually in line, because of course, if you have something and you have a table with like 50,000 lines, there might be small things, there might be exchanges, and this is actually checked. And the good thing is, once it's in the database, you can actually feel that it has been checked and is good. And we had a data management plan, including data licenses. So one can actually give general public access to the data. The data is structured according to this, and partners are informed. It's transparent, restricted access where needed. So Germinate was actually adapted for our berry fruits because we had to introduce sensory profiles, as I mentioned. That's very important. Consumer studies, which were not yet implemented in the Germinate platform, and you just see some of these traits. So new template files and new visualizations have been implemented, and we will see some of this work very soon. So the work has been done by Elisa, Paul, and Sebastian. The latter two at the James Hutton Institute in Scotland, as Bruno mentioned, and we see, of course, funded by the European Union, and would not have been possible without all the partners that contributed here. And with this, I would like to hand over to the actual demonstration, and just to give a brief introduction, what had to be done and where we are now, that we could actually reach this nice stage that we are in now.
Right, thank you, Björn. Let me share my screen. Right, I'm going to assume that you can see that. If not, please just let me know. Right, so I'll be talking about Germinate itself then, specifically how we're using Germinate to make the breeding value berry data available. Hang on. There you go. So first of all, who we are, Sebastian. I'm Sebastian. Paul, you see there in the picture, he's also in the audience. We both work for the James Hutton Institute, which is co-located with the International Barley Hub and the Advanced Plant Growth Centre as well, just outside of Dundee in Scotland. You can see our shiny new building there on the left-hand side. Now the work that we do, Paul and I specifically, is we are finding software solutions for biological challenges. And that can be anything from data collection to data management and then data visualization as well. You can see a few of the different tools we've developed over the last years here. Now the one we'll be looking at today is Germinate, but you can see that it interconnects with these other tools for data collection and visualization. So it's kind of a tool chain of things that we have in place. But this presentation is just about Germinate. So if we then think about what Germinate is, we often use a slide like this one. So you can think of Germinate as this plant. And there are various different things or various different data types that are available in a project like this one. So you have things that were already mentioned by Björn. So you have passport data, genotypic data, trials data, but also things like pedigree, sensory data, and images. Now, these are generated by different partners, so only specific people will have access to them. But if you want to make them available to everyone else within the project and outside the project as well, there needs to be some centralized database in place. Because as you see it here just now, the data is kind of below ground, so it is there, but nobody has got access to it. So basically what Germinate does then is it takes all of these different data types and integrates them into a central repository, which is the plant itself, basically. While it does that, during the data import, it checks the data to just make sure that it's consistent. It flags issues that often happen during data collection or things that might look a bit wonky, so that you can go back and look over it again. Now, once you have that central repository, it's quite easy to share that with the project partners or collaborators or the stakeholders, but also to make it available more publicly to the general public as well. Now, sometimes that's not quite possible. So sometimes some of the data or all of the data needs to be kept private. And that might be for a certain time period, or it might be forever. In this case, most of the data is public. There are only a few data sets that need to be kept private for now, and that's only until there has been time to analyze the data and to publish the data. Now, on top of all that, there needs to be ways in which you can interrogate the data, query it, and filter it to find the things that you are interested in. So, for example, if you're looking for trials that are looking at different chemical treatments, or are looking into drought resistance or anything like that, you can use Germinate to filter the data down to just those data sets that you're looking for. Now, once you've selected the things that you want to find, there are various visualizations, and we'll see quite a few examples on the next slides, in which you can visualize that data. And visualizations are fantastic because they give you a general overview over the data, so you can get an idea of what data there is. They're also very good at highlighting potential patterns or data outliers as well. And then you can drill down into the actual data points and then look at those potential outliers, for example. So basically what you're trying to do with Germinate is take you from this situation where you've got your data in various different versions. One of them is on your laptop, one of them is on your hard drive, one of them is on the cloud. One of them is sent to a colleague, they made changes, and in the meantime you made changes. And it's a nightmare, right? We all know this. Nobody wants to do this. Nobody wants to manage their data that way. And what we want instead is what I just presented. You want a central location where all the data is hosted, and then everyone knows where to go to get that data, and you can upload new versions as they become available. Now, before we go on, I thought it'd be kind of interesting to see how many of you have already used Germinate in the past, or how many of you have heard of Germinate. And for that, I'm going to try and find the chat as well, which I can't on WebEx. But if you scan the QR code here, you get to this poll, or if you go to slido.com and enter this number, you get to the poll as well. And then we'll give it a minute or so for some responses to come in. Okay. The poll will be active after this as well. So if you haven't had a chance yet to submit your response, you can still do that. So it's interesting to see more than half have actually heard of it but not used it. Then about 40% haven't even heard of it yet, and then there's a few who've heard of it and used it. So hopefully after this presentation, that last number is going to increase significantly. So if I move, hang on, if I move on. So Germinate isn't just used by BreedingValue. There are various different international projects that use Germinate, and you can see some of our collaborators in the top right here next to BreedingValue. So we do a lot of work with the Crop Trust. We've worked in the past a lot with Stimmit as well in Mexico, and we've also done work with KAUST, so that's the King Abdullah University of Science and Technology. And the crops that we've covered, there's a few examples here. This is not all of them. I couldn't fit all of them on the slides. The various different things, like obviously barley, because we work with barley at the Institute, also softfruit. We have an existing softfruit database unrelated to BreedingValue. There's some potato data, then maize and wheat at Summit, and then various different less known crops like finger millet, pearl millet. There's also chickpea and sunflower and various interesting things like that. Another quick poll, and this is the last one of the presentation, just to get an idea of what is the main crop that you work with. If you could just respond with, the short English name, because this will generate a word cloud, if I remember correctly. Okay, so that's quite interesting. So there's a lot of variety there. Obviously, lots of berries, strawberry being the main one, then raspberry, and then lots of different other berries like honeyberry, there's blackcurrants, blueberries, lots of different ones here. Interesting. Right. Okay, so is that the next slide? Yeah. So if we look at the data flow within the project, and Björn and Elisa mentioned that already on their slides, there's all of these different data types that are being generated by the different project partners, as we see along the top here, and they all need to go into Germinate. And the way this was done is using the data templates that we all decided on, and that data was submitted to the Forschungszentrum Jülich, where Elisa looked over the data and made sure that everything's in the right format, everything's consistent, everyone's using the same trait names and germplasm identifiers. And that's a huge amount of work that went into that. Because only when the data has been cleaned can we actually upload the data to Germinate. And for that, we are only using the data templates that we provided for Germinate. So even if data was submitted in other formats, it had to be reformatted into those data templates so that we can upload it automatically to Germinate. That work was done at the James Hutton Institute. Now, once it's in Germinate, that data is available to the general public, or some of the data is available to the general public, and I'll talk about that in a second as well, so that you can go and explore it. And at the end of the presentation, there's a link that you can just follow to explore it yourselves. So just a quick disclaimer, because I thought this was important. So Paul and I, we are experts on Germinate because it's our tool. We develop it. We know everything about Germinate. So if you have got questions about Germinate, great, because we can answer those. If you've got questions about the BreedingValue data specifically, we probably won't be able to answer those. So in that case, you probably want to talk to the project coordinators or the project partners who generated the data. So I will be showing data and I will give you my interpretation of the data, but take that with a grain of salt, because I'm not a berry breeder. So if we look at the passport data first, now that, if you're not familiar with the term passport data, that includes things like germplasm identifiers, synonyms, taxonomies, but also the pedigree or the biological status. And that's just whether it's a breeder's line or an advanced cultivar or anything else. And in some cases, that might include the country of origin if it has been collected in the wild. And that information is absolutely essential to provide context to the other data types. Because if you see phenotypic data or genotypic data with just an identifier assigned to it, but you don't know anything about that identifier, that data is basically useless because you can't relate it back to the actual material that was used. So passport data, extremely essential. And we'll have a look at the passport data now. So the first breakdown is just by taxonomy. So we see a visualization that's from Germinate here on the right-hand side, which is a so-called sunburst diagram, where in the innermost level, you've got a breakdown by genus, then it's by species, and then we see some subspecies here as well. And most of the data we have in the database is about strawberries, and I think that's due to one very large historic data set that we've got from UPM. It's a 20-year historic data set, and I think that's why the majority of the taxonomy is just about strawberries. We see the largest breakdown into the different species here as well, also in strawberry, and then we've got raspberries as the second biggest section, and then some blueberries. It's the smallest proportion in the data set here. Now, if we look at the breakdown by biological status, and that's based on the FAO terms, based on the multi-crop passport descriptor biological status, you can see how it breaks down into the different categories. So most of the material are breeding or research material, followed by breeders' lines. And then about 10% are advanced or improved cultivars. And then there's some few hybrids or wild germplasm entries here as well. Now, within BreedingValue, there was another type of classification which isn't the FAO MCPD, but there was another type of another way of breaking it down by material type, which we see here on the right-hand side. This shows the percentage per species or per genus here, actually. That's wrong. You can see for strawberry that is mostly pre-breeding or advanced selections. And for blueberry, it's mostly undefined, other cultivars pre-breeding or modern cultivars. And for raspberry, we see mostly undefined or it's population material that we have in the database. Now there's a very useful index indicator that's being used for measuring the completeness of passport data, which is called the Passport Data Completeness Index, published by Theo van Hinten in 2011, which ranges from 0 to 10. 10 means there's a perfect set of passport data. All the data could be there, is actually there. We can see that there are a few here that have got very good passport data completeness, but there's a huge chunk of data that doesn't have a lot of passport information. And I assume that's due to privacy concerns by some of the partners who just didn't want to submit that data. Now, if you break it down by project partner, and I don't expect you to actually read or be able to read what's in that table, it just shows all the different partners and the data sets that they were involved in. And if you look at this on Germinate, you can actually then explore the data sets from a particular partner. You can see which ones have generated the most trials data, which ones have generated the most genotypic data, and so on. And this is just to give credit to all the project partners to show what data they generated so that everyone is attributed appropriately. This is, I should mention this is work in progress. We're still uploading data. There's already a good chunk of data there, but in the next few months, we'll upload a lot more data. So that'll become available very soon. So I mentioned earlier that, sometimes you need to restrict access to data. That might be to keep it back from publication, or it might just be that you want that data to be available under certain conditions. For that, we usually use Creative Commons licenses because they're very standard, they're very well known, and they come in different flavors. So most of them require attribution to be given to the author. And that's the first one you see here. We've got 14 data sets that require a license like that. Now, in addition to that, you can request that anyone using that data has to publish their data using the same license terms. which is the second one here. 8 data sets require that. And then we've got another clause here that says it can't be used for commercial work. It can only be used for non-commercial work uses. There are 8 data sets that require that. There are 9 that require consultation first before the data can be used. So in those cases, you need to get in touch with the project partner who generated the data first, and only then can you use the data. And 2 of them are currently not public because they're held back until we've had time to work on the data and a publication has been prepared. Right, so let's look at some actual data then. If we start with field trials data, this allows phenotypic comparison of germplasm. You can compare the performance of an individual against the average. There's things that let you assess the performance across different sites or across different years. And the first example here that we have is the 20-year data set from UPM. And the visualization you see is the so-called scatter matrix. Now that just means that you've got different scatter plots. Each one of these blocks here is an individual scatter plot, and you've got the x-axis description here and the y-axis description there. The scatter matrix is very good because it shows you the relation of the different trait combinations across a big selection of traits. And we've colored this here by year, so you can see how the data evolves over time, or how data might be slightly different in a specific year, and so on. But if we just zoom into one of these charts, you can see this one here, for example, which looks at surface color, and it looks at 2 of the surface color variables. This one's A, this one's C. You can see that for all the years there's a very linear correlation between those 2 variables. But what you can also see is that there are some data points that fall outside of that main data distribution, and those are mainly from the years 2020 and 2021. Now again, why that's the case, I can't explain. I can just show you that what kind of visualizations Germinate can generate and how it's easy to spot those interesting data points. Because if you were just looking at the data in Excel, it would be difficult to find them that easily. If you're looking at the one to the left of it, again, there's a nice correlation between those two traits that we see here. This time we're looking at L and C of the surface color. But again, there's a couple of data points, or a few data points over here that kind of fall outside of that data distribution. If we look at development over time, I found these 2 examples which I thought were quite interesting. So left-hand side shows the total yields versus the wasted fruits per plant. And if we just look at a few example years here, and these are just lines that I drew by hand, just to show you kind of the trend per year. So you can see that in 2002, 2004, we've got higher number of wasted fruits per plant. Whereas the later years, 2020 and 2021, that value is a lot lower. Interestingly, though, on the x-axis, the total yield in those 2 years started a lot earlier, a lot lower actually. Whereas 2002, 2004, the lowest total yield values kind of end here and there. So an interesting development going on there over time. The right-hand side, we're looking at antioxidant capacity versus phenolic content. As far as I understand, those are both good for human health, so you would want those values to be higher, I assume. And if we're looking at some representative years here, for example, 2006, we've got very high values in phenolic content, but with lower antioxidant capacity, whereas 2016 kind of flips that. So you've got higher values in antioxidant capacity, but lower values in phenolic content, and 2002 kind of sits in the middle there. So Germinate gives you a lot of ways of looking at the data and just visualizing it. You can at any point click on any of these data points to find out what germplasm was used to generate that. You can select certain data points in here to add them to your download list, for example, if you want to download the genotypic data for those various ways in which you can interact with these charts. Another example, this time blueberry from the James Hutton Institute, and the same kind of visualization, a scatter matrix of different traits that we see here. And this one I found quite interesting, which is looking at the average single fruit weight on the x-axis by fruit size, And this is again colored by year, this time only 4 years, because for the project duration. You can kind of see how it's a fairly linear correlation between those two, but it kind of jumps up in the fruit size each year. And when I was talking to Susan McCallum, she was saying that that's the expected behavior. You would expect the fruits to get bigger over the years. But that's interesting to see in a visualization like that here as well. Now, when data is collected over time during the season as well, that automatically gives you time series data. Now, time series data, there are different ways in which you can visualize that. And we're going to look at some examples here, because they give you a visualization of the development of the values over the season. For example, you can identify whether a significant event has taken place, like certain weather conditions, for example. You can also compare the effect of treatments over time. Now, this is blueberry data again from, I'm going to pronounce this wrong, the Kukurova University and also Alex. And we've got 4 different traits or measurements that were taken here over time. And they're looking at two blueberry varieties here, Biloxi and Star, with 2 different treatments. So one has been treated with GABA, the other one with hexanal, and then there's a control, obviously. So the blue ones are Biloxi, the reddish ones are Star, and you can then see the development over time for those selections that were made here. If you're looking at the firmness, for example, you can see that the controls, which are the red and the blue, got the lowest value over time, and then you've got the treated ones have got higher value when it comes to the firmness. The gray line, by the way, is the average across all the data, not just the selected germplasm that I'm showing here. Similar sort of story for chilling injury of fruit. You can see that the controls have got the highest percentage of chilling injury, and the treated ones are kind of below the average here. This one I found quite interesting as well, because you can clearly see that the reddish ones are generally lower than the bluish ones. So you can see that Star is performing, well, I can't say if it's better or worse. It's got lower values across the board than Biloxi. But again, within each germplasm, you can see that the control is always the lowest, and then you've got the different treatments which affect the phenotypic value here. Another type of data that we store are environmental data points. These will be climatic things like, for example, temperatures, rainfall, humidity. There could be light measurements as well. And they will be collected sometimes across years. You can see how that develops over time. This is one example here from Junta de Andalucia, where we're looking at different environmental measurements. Again, same kind of visualization as for the trait data. You can plot all these environmental variables against each other. And then if you zoom into one of these, you can see how the 2 traits relate to each other over the years here again. Interestingly, you can see, for example, in 2023, the maximum humidity ends somewhere here. So the lowest value is actually 49 or so, whereas in the previous year, there was lower maximum humidity. If we're looking at a different one here, this is showing average temperature across the year for the 3 years that were selected. And you see a very similar shape here, because I mean, you would expect the temperature to go up in summer and then back down for winter. But if you look at the purple ones, for example, you can see there's a lot of variation across the year. So it starts quite high, then goes down, back up, back down, and so on. Whereas in 2021, the value started a lot lower. So again, nice visualization just to give you ways of looking at data over time, looking at different trait combinations, and seeing the effect on, for example, treatments as well. Another environmental data set from UPM here. If we zoom into this one, for example, we see the minimum daily air temperature versus the potential evapotranspiration. And again, across 2 years, you can see there's a bit of a shift going on here. So 2021, lower minimum daily air temperature across the board, and slightly, well, similar to slightly higher potential evapotranspiration there. And you can see the breakdown of the different years here in the different environmental variable combinations. Another data type that we work with are images. Now these give you detailed images of individual germplasm, for example, and they can be categorized using tags. These tags are then searchable, as well as the germplasm is searchable as well, based on those images. What that looks like then is, at the moment, we have got 254 images. I think they're more ready to be uploaded. But you can see those ones here from Hansa Bread. Obviously all strawberries, but they have been tagged in with 3 different tags. So there are images that show cross sections, there are images that show the whole fruit, and then there are images that show the punnet, which we see here in this example there. And if you click on any of these tags, it will filter it down. In this case, I've clicked on cross section, so I'm only shown cross sections of the individual fruit. And then you can click on that to look at the image in much more detail. Now that's the generic image page, which just shows you all the images. If you're just interested in the images for a particular germplasm entry, you can go to its passport page. And that passport page has got lots of different sections here, but we're looking at the images here at the moment. So those are all the images associated with a particular germplasm entry. And you can then, similarly to before, you can filter these down, or you can look at these images in more detail. Now, the information about the images is also available on the main germplasm table, which we see here. So that's the one that just shows you all the entries that we have in the database. Now, these have been filtered to only show the ones for which we have images. In this case, there are 44 germplasm entries for which we have images. And you can see the individual image count here. And if you then click on that, you will be able to go through all the individual images. You can also see here that for all of these, we also have trials data or phenotypic data, and we also have some pedigree data for some individuals in this selection here. Yeah, so the next type of data was something new that we hadn't worked with before. It's sensory type data or consumer preference data. And that gives you information about things like preference or on taste, smell, mouthfeel, and visuals, and that kind of thing. And for that, we added a new type of visualization, which are these spider or radar charts, which basically they're just parallel coordinates on a radial arrangement. So you've got these different characteristics or variables. Each one has its own axis here or parallel coordinate. And you can then see the value for, in this case, the responses from 2 consumers for 1 germplasm across these different variables. And that's then called the so-called profile. So these kind of charts allow you, in this case, for example, to look at the responses from different consumers for the same germplasm. But there are also versions which I will show where you can compare the performance of different germplasm across these selected variables. Let's just look at an example from strawberry Hansa Bread data. We're looking at this particular germplasm there, and I've searched for variables that contain visual, and then I've selected these, and that gives you the profile of this germplasm for those visual variables. And you can see that the value is fairly high across most of them. Just the willingness to pay based on the visual appearance was quite low. So I assume that they had quite a high price. Might be the reason for that. Looking at a different example here, this is again strawberry from UPM and CNR. Now in this case, we're looking at the responses from 5 different consumers for the same germplasm here, Francesca. And some variables that are selected here, and these are kind of overlaid on top of each other, so you can compare the responses from the individual consumers. So, for example, the orange one scored it a lot lower than, for example, the blue one, and the gray in the background is actually the average across the whole data set. So you can see, for example, I don't know what some of these are. The firmness of the texture, the purple and green scored quite high. Whereas purple scored very low for the visual color appearance. If they're shown on top of each other, it makes it kind of difficult to see some of the ones that are further down. So you can always split them into the individuals, and then these individual consumer responses will be compared against the average. So you can see this consumer could score everything lower than the average across the board, whereas this one was kind of happy with it, except for these two variables here, the color visual and the aqueous appearance visual. Right, last type of data that I'm going to show here is genotypic data, and that's usually when we deal with genotypic data in the context of Germinate, that's SNP type data. So you've got a marker and you've got a location, and there's a call assigned to that. And this kind of data allows you to look at the genotypic differences between germplasm, and there are various ways in which you can export that data to visualization tools. For example, Flapjack, which I'm going to show you now as well. So the way you would export genotypic data is, first of all, you select the data set that you're interested in. In this case, we're looking at raspberry data from JHI. It's 2023 SNP marker data. And if you just select that, it gives you options in which you can subset the data. Now, for example, if you had selected data points in one of these phenotypic data charts, you selected some data points in there, and you want to export the genotypic data for those, you could use the list or the group feature of Germinate to export data against those. Now, the same can be done for markers. If you're only interested in specific regions of markers rather than everything, you could subselect the markers here as well. Now, in this case, I just exported everything. Further down, there are different export formats that you can select. In this case, I just wanted everything. But there's things like HubMap, which is a common format for this type of data. Flapjack, which is the tool developed at the James Hutton Institute as well, which we're going to look at now. But the export is just going to contain everything. And once you hit export, that's going to pop open the side menu that tells you, some data is exporting. Then that'll go away and export the data for you. And then at some point that'll be complete and you can download the data, which will be zipped into a zip file, which contains all the data files that I requested. So these are the flat data files, that's the HubMap file, and then we've got the Flapjack file here as well. Now, if you have Flapjack installed, you can then just double click the Flapjack file and that'll open your data in there. And then that looks something like this. I'm not the developer or an expert on Flapjack, so I'm not going to explain how Flapjack works. But basically, it's a genotypic data visualization tool, so you can look at the individual calls for the markers and the germplasm. It does include some analysis features as well, but you need to have a look at the documentation for that. Right, so that kind of wraps up the data overview. Just as a bit of a summary, if you're interested in Germinate, there's a link or a QR code there where you can look at more information. I'd like to highlight that it's completely free to use. It's completely open source, so whatever you want to do with it, you're free to do that. If you would like it for any of your other projects, please get in touch with us. We're more than happy to give you a demonstration session or to get you up and running with your project. That's not a problem at all. Just get in touch. Now, the BreedingValue data specifically is available at the link you see there. There's also a QR code that you can scan. Now, not all the data is there yet. Not all the data is public yet. But there's a lot there already for you to explore. You don't need to create a user account or go through registration or anything. The data is just there for you to use. Some of it will require you to accept the license terms that I mentioned earlier. Some of them will require you to get in touch with the project partners who generated the data as well. But there's information about that on the website itself. Some quick acknowledgements. A big thank you to Elisa, who did a lot of the data, getting the data into a lot of the work, getting the data into the state where we could actually upload it. Also, a big thanks to all the BreedingValue partners who sent us their data, and especially those who agreed that their data can be made public. And obviously also the funders of the BreedingValue project, but also all the funders of our continued Germinate development. That's the last slide.
Thank you very much, Sebastian. Well, for sure, demonstrate that we have generated many data, so that it's really an important work of the project. I don't see a question at the moment on the chat. I just, I don't know, just to promote eventually possible some question. Well, I have I have note to three things. For example, the passport data. Do you think that that is something that has been discussed, I think, already in the group? But the passport data, is this database a source of information that can be of help also for the other international organizations that in some way are dealing with plant variety rights, privacy registration or patenting and so on. Do you see some potential lead or support to the activities that such as an organization are doing? That was discussion yet some times ago, but in reality now we can give some more details.
I'll leave that question to Björn or Paul to respond to.
Yeah. I don't know. That's I see that there are some other people from the organization that are following the seminar. So maybe the question can be addressed also to Lorenzo or to somebody else that I saw that.
Björn, hi, this is Elisa.
Yeah.
I maybe state a brief comment on that. Of course, our data that we provide on Germinate might be used by others freely. However, for especially passport data, I must also admit that I was struggling to find sound or conclusive passport data for a specific accession, even in other databases that are specified on passport data. Because there are usually multiple entries. So for one accession, you might find 5 or even 20 or 50 entries. Many of them might differ. Some might have more data than others. So it's quite difficult sometimes to find the correct information. It's also possible that there are mistakes in these databases because it's usually not manually curated. So this is a very difficult question, and of course we do the most we can to only provide, of course, the correct data and the most complete data we could find. And especially the Julius Kühn Institute was of great help for the strawberry accessions that we have in our project. And for the other two species, blueberry and raspberry, we also had great help from the James Hutton colleagues. And also from the Turkish partners or Italian partners that work with the two species.
Thank you, Elisa. On the chat, there was a question, do you have a REST API exposed for Germinate? Sebastian answered, yes, Germinate supported the breeding API as well. Yeah, so in fact, that was, I don't know, you already make many examples. If I have a small company that I'm doing breeding, what can be the benefit to be in contact with you? So how to look at all this data? So again, just try to involve more people that follow it.
I think a good demonstration was already given by Sebastian when he showed, especially that you see certain things which you couldn't see just in a table, which brings back, of course, that this interlinking is quite nice and the interactivity, which helps one to explore and actually maybe find some germplasm, which is of particular interest, which one would have not found like this.
Okay. In the meantime, if you follow the chart, You can also speak instead of, of only writing. So Lorenzo Maggioni is asking how far this is open and create a link and connection with Eurisco. Genesis is another network. I think that could be very, very interesting.
So do you want me to answer about Eurisco and Genesis? So for people who don't know, so Eurisco and Genesis are, we work with them both. We have a book chapter from last year published with them as well. They're what we deem kind of germplasm catalogues. So Eurisco is the European version, and Genesis is the worldwide version, which also encompasses most of the stuff that's in Eurisco, although not all of it, as far as I'm aware. Very easy to have links from Germinate to Eurisco and Genesis, and in fact we do that. We've not got it with this project as far as I'm aware, but we do it with other projects with the Crop Trust. We work really closely with them. And we also have, I mean, internally, we've got potato collections and everything gets deposited. We've got tools that export the data from Germinate in the correct format for Eurisco and that kind of thing. So yes, absolutely, it's important we have links to them. I think the main differentiation between those catalogues, so Eurisco and Genesis, we describe them as, I think, wide and shallow. So they have a lot of different germplasm from a lot of different crops. But they're phenotypic and genotypic and background. They've got passport data, but other background types of data that we store, they don't tend to have that in such detail. Whereas we have a more narrower and deeper data. So it's more narrow in scope of germplasm, but much deeper in terms of the richness and complexity of the data that it's holding. And they both exist. They're both useful with each other. We have cross links between each other as well. They're all part of the FAO's GLIS system.
Thank you, Paul. So, okay, then the discussion about the happy connection. Well, then I have another. When you made the interesting animation, so interacting with the people, asking about the different type of interest of crops and so on, it turned out that strawberry clearly was more important than raspberry. Even blackberry, it was probably more important than blueberry. Well, and actually, in the project, we also had this fact that there are probably very few partners that really involved on blueberry. Actually, the most important is James Hutton. The fact is, that in the EU, the importance of blueberry is increasing really a lot. So I think that the project has a lack, all Europe has a lack in terms of knowledge on blueberry. So probably that is something that, I don't know, from your point of view, what if you have a comment on this concept.
So can I pick up on the point, Bruno? If I understood, correct me if I'm wrong. You were talking about when Sebastian put up the Slido asking people to put in the crops that they're interested in, and there wasn't so much blueberry. I think we need to go back and look at that because, if I remember correctly, I think there was some spelling errors in people writing blueberry. So it was maybe the impression was maybe false, but we have that data and we can share that amongst our consortium. The response to that. So we should be a bit careful because it probably was quite well represented, but we can check that.
Yeah, however, it's something that we should consider because, again, in the project, blueberry, it was present, it was active, but probably maybe for the next future, we should get more in deep on the possibility to expand research and knowledge on blueberry, because otherwise it would be more difficult because a lot of interest is on blueberry and everything is coming probably from, most of the material is coming from outside Europe, so that it's again not really positive. I don't see other question. But take time now to don't be shy if somebody
Hello?
wants to say. Hello, yes, please.
I have a question. I don't know if you can see me.
Not yet, sir.
Okay, maybe I have a problem to turn on the camera. Let me see.
No, we did. You are David. David Pierre.
Yes,
Okay.
yes, but I cannot turn on the camera. So I am David Pierre. I'm working for Honestom database, Honestom project. First of all, I really want to thank you for this work, this wonderful work you are doing. And I have some questions. I have like two or four questions. So what I understood from the presentation is that for the data to be into your database, people need to submit the data to you or to send the data to you like personally.
Well, no, that's just the way we did it for this project. So we can define data curators as well. And in that case, the data curators are able to upload the data to Germinate. So it
Yes, I
doesn't have to
understood
be
that.
us.
Okay, for example, myself as a public user, I don't have any account because no registration is needed to use the database, the platform. If I have my own data and I want to submit the data, like I want to put my data into your database, how can I do so?
For this project, I guess you'd have to talk to the project coordinators because I don't know what the plans are for the database after the end of the project. So maybe Bruno can answer that.
I think I was going to just follow up on what Sebastian's saying as well. As I mean, I think I get what you're getting at, David. You're asking how you, as just a normal user, compare your data, and this may sound bad, but really we don't want that happening in that way. There's a huge issue if you let everybody add their own data. There's a huge data quality issue, and it's massive. And I don't think that's fully appreciated sometimes. So what we tend to do is have contact points, somebody who knows about the data set, and it could be that's you. I mean, that you could be the person, absolutely. But it has to come through some sort of curation system. And under this project, that work has been done by Elisa, and then data's passed on to Sebastian. And I mean, I share an office with Sebastian, and I can assure you there's been lots of pulling out of hair and things, finding problems, and you find problems down the line as well. No matter how hard you try, you're never going to get them all in one go. So we absolutely shy away from letting people add their own data unless it comes through a data coordinator.
Okay, I really understood that because what I got is that like the person needs to contact like a data curator or like a contact point and see the data, and after you will do maybe some data processing to make sure the data is completely curated before you can like introduce the data into the database.
Yeah, I will say there's never completely curated data. That doesn't
Yes.
exist. But as much
Okay.
as we possibly can, yeah.
Okay, so as this system is a BRAPI compliant system, can you exchange data with other BRAPI compliant systems?
Yeah, I'll let Sebastian answer that. That's right up his
Yeah, I
street.
mean, that very much depends on what you mean specifically.
Okay, let's say myself, I'm working on Honestom database. On Honestom, mainly we store crops, we store tomato accessions data, but Honestom can also store other data, other crops. So we can exchange data. Let's say in Honestom, I want to add data for strawberries. Can I exchange data with you?
Again, I'm not entirely clear what you mean specifically. I mean, the breeding API, there's documentation on which endpoints we've got implemented. So you can pull data from Germinate through the breeding API. If you've got an account, you can also, if it's enabled, you can also push data into Germinate. So it depends which way around you want to exchange data. But yeah, we can. If you want to send me an email afterwards, we can have a chat about that as well.
Yeah.
Yes,
Okay,
Sebastian, there's also examples of using like tools with BRAPI. So we can pull data from Germinate using BRAPI with tools like Sebastian's going to correct me if I'm wrong, Flapjack and Helium for pedigree visualization. So we do have, and potentially in the future other things that are coming up. So we do have very good BRAPI support, and we're I guess core members of that BRAPI team as well, and we contribute to the project.
Yeah, can I say something? I know that
Yes, please.
Arneston is practically a parallel project of BreedingValue. It has been funded with the same scheme of the Horizon 2020 program. And we, since the beginning, with Tony Grenard, that is coordinator at Arneston, we were trying to keep a connection and to develop something together. Tony was even invited to participate in some of our meetings. So I think that it's good that you find an interest in the work that we have done in this, in creating the BreedingValue database, BreedingValue database into Germinate platform. I think that it's really interesting. Maybe we should, and it's very positive also for the Horizon 2020. We are both ending the project. I know that your project is ending in December, we are ending in June. And our reviewers already point out the fact that we should try to create some connection between project, actually among projects, because there is also a third project on olive oil that has been funded in the same scheme. So this issue can be taken in a discussion that we can have probably among us, involving Tony, involving maybe also the coordinator or coordinator of the olive project, that is also another very big project, and see if we can really create a link for all this type of information, a cross-link or a cross-exchange of all this type of information. And I will say that the Commission will appreciate a lot this type of work if we can manage.
Okay,
so I will keep really open this discussion for the next period.
Okay, thank you so much.
Yeah, well, thanks. That is a very interesting idea, and very, in my opinion, if we can do something, it would be very helpful for all of us.
Sure.
Well, any other question?
No, it's okay for me. It's okay for me for now.
Okay, then. Yeah, let's stay in touch. Speak with Tony. I saw that Tony was connected before. I don't know if we're still connected, but maybe you can discuss with him and set up a meeting together so to discuss the future possibility. Another is Chris Norman. It's just now posted in the chat. Chris, do you want to say something? Yes, just asking about, is it possible to work through download this deep data in a flapjack format? I'm struggling to download any useful information about the available markers. Is somebody able to answer to this question about the genetic information?
Should I just show that again? I can do it on the live website as well if that's useful. Hang on. Share that one. Right. So if you get to the BreedingValue website, there's a section on the left-hand side that says data, then there's genotypic data, and then there's genotypic export underneath. So you kind of expand them like that, and that's where you want to go. There's one data set publicly available here just now, and that requires you to get in touch with people at EPM first before you can use that. After you've done that, you can accept the license terms there. And once that is done, you just select the data set there, pick the flapjack export option, unless you want to subset it further by groups, which you can obviously do up here as well. But if I don't do that there, hit export, it will go away and start exporting that. That'll take a few seconds or so. I can't remember how big the data set is. So let's leave that running for a little bit. This is quite boring to watch now. But basically, when this is done, it'll download the zip file that I mentioned on the presentation slides, which will have the flapjack file in it. So that's the download done. Saving that. And then when I open, where did that go? Open. Oh, it's on the other screen. Hang on. That, right. Once I've opened that, there's the flapjack file there. So it's really just, clicking through the interface from the genotypic data set, picking flapjack, and then hitting export. And that's all you need to do. And if I open that here, that's going to look something like that. Okay, hope that clears it up.
Sorry to interrupt again. May I ask a technical question regarding Germinate servers? May I ask where the data actually go to when I upload something there?
The data is currently held on our institute servers here at the James Hutton Institute. That can be moved to any other place if required, because it's quite, it's an easy setup to just move anywhere else. But at the moment, the data is held here at the James Hutton Institute.
Thank you.
Okay. Zach. Well, thank you very much. I hope really you appreciate the presentation and the discussion. It was very interesting for me. I say everything has been recorded, and in any case, it will be available on the web page of the project. So for other people that next want to follow it, and to know about the Germinate platform, really feel free to contact myself as a coordinator of the project. But again, for more specific question about Germinate, Paul and Sebastian, about the data management, Bjorn and Elisa, that are the responsible of all data collection and management for the project. And again, if nobody wants to have additional question or requests, I want to share you again the screen, just remembering again that we have now the final workshop expected next April 2 and 3, so that is the date here. It will be in Ancona. It will be in presence, but for sure we will provide also online connection. So if you will not be able to join us in Italy, you will be able to connect and follow our activity and have more larger view of all the different experimental trials, genotyping and phenotyping tools that we have developed and results that we have developed, and clearly also genetic material that we have identified of interest for the different characteristics related to sustainability and quality for the consumer. So really, I look forward to maybe meet you in person next time, and that will be and at that event eventually does have a connection with the other project, Armstrong, maybe also the Olive project. I will try to create a connection through the coordinator and see if we can share some of this information related also to the other two projects that have been funded. But this really very interesting program that was included in the Horizon 2020 program. So I say goodbye to everybody, and thank you to all my colleagues that have done the work and given the presentation, and all you as a participant that decided to follow the noises and contributing positively to the success of this event. Thank you very much to everybody, and bye-bye.